One of the things we're doing is that all of this stuff is ending up, well, it ends up Kruger's one of these world sites where people from all over the world come to see wildlife. It's still one of these amazing sites. And so there are lots of villages just outside. So all of this water is running by villages, and people are still taking buckets of water down and pulling water out of the, the rivers, and that's what goes home with them. So it's a very different scenario than what we have here. Now, is that everybody? Of course not. But there is still a proportion there. So it goes back to this idea that we talked about a number of years ago about the concept of a sentinel species, a species that can actually maybe tell us something about the health of an ecosystem, the health of a river system, the health of other wildlife that are there, the health of the people that are actually using that same system. And so when the die-off of crocodiles took place, they also had a major die-off of the catfish. So then the question is, well, why did catfish and crocs die, but not tilapia, which are actually native to there, right? Well, is it because of an appropriate, top of the food chain or association with a sediment? Could it be metals, toxin? We don't know, and that's what we're trying to do. There was a national call. This was the second round. So the initial round, if you remember, after the Deep Horizon oil spill, there was a demand that BP put up a significant amount of money into a trust fund that would actually support research. And then, of course, there was separate money for economic development and all that. But this is for research to try and understand both the short and long-term consequences of this oil coming into the Gulf. And so the initial round of grants largely went to the states that were directly affected. So they were the Gulf states. The second round, they were doing somewhat the same thing, but they also said we're willing to take grant proposals from anyone in the United States who have interesting ideas. So one of the things that Dimitri Spiropoulos, John Botts, Satomi Kono, and I have been doing is asking questions about how do you, in fact, screen chemicals in a way, okay, that you don't have to test a whole bunch of live animals to do that. And because, to be honest, there's two things going on here. The first is there's a large number of chemicals out there that we're exposed to. So there's no way, I don't care if you gave me $500 million, you gave me the whole trust account, okay? There's no way in my professional lifetime I can do every possible combination, even if I use environmentally relevant concentrations. So forget screaming levels or little tiny levels. Even if I just go out and say, okay, I measured all of these chemicals in these dose ranges, doing the combinations of animals, what animals would potentially be exposed to, we can't do that. So how do we do that? Well, you can't, first decision you have to make is that you can't solve all the problems with one experiment. Each one of us know you got to chip away at the block, okay? And some of those chips are big, and some of them are just trying to figure out, what's the sculpture inside the rock. So we actually do a couple of things really well as a group. We have worked extensively for years trying to understand how environmental contaminants and endogenous hormones, native hormones, influence gene expression via steroid receptors. So mimics of estrogen, mimics of progesterone, mimics of testosterone. And we actually have a technique that we've developed, in fact it's a couple of different assays, where we can actually take the estrogen receptor or the testosterone receptor, this is the receiver, okay, from any species that we've cloned down, and we've done 40 or 50 species, put it into a cell, put a little reporter construct in there, so that when we add a chemical, goes into the cell, binds to the receptor from whatever species we're interested, that then is translated over to the little reporter, binds to the reporter, turns on a gene, and the cell glows. And it does it in a dose-dependent fashion. So now you can say this chemical can be an estrogen, an androgen, a progestin, whatever, okay? What the dose is, so is it relevant to what potentially a cell would be exposed to? And does it say that that's now a problem? Of course not, but at least gives you the first stop of saying, okay, we now know we have an active compound. Out of the 100 we tested, we now identified 1 or 2 or 3 that we think are really important. A stem cell is an undifferentiated cell, right? It's a cell that potentially could become many other cells. depending upon the information it gets during that developmental process. So in a developing embryo, certain signals come and say, you're going to be a fat cell. No, you're going to be a muscle cell. No, you're going to be connective tissue. No, you're going to be bone. Can we also take this thing called aged oil or whatever thing we're interested in and using several chemical techniques, fractionate that into little fractions and again, test them and say, okay, these cells are supposed to become muscle cells or connective tissue. But we put it on and it becomes a fat cell. There's a whole world now where we're starting to realize there are compounds called obesogens. So these are these are chemicals that in the developing embryo, instead of stimulating the production of muscle or fibroblast cells, it actually stimulates more fat cells. So this is actually a growing area of saying this obesity epidemic we have is clearly associated with poor choices in food, especially in children, let's say. But it's also, and just having availability of calories, right? But it's also associated with, we may be programming during embryonic development that body to store more fat. Several chemicals now, which are common, things like BPA, which comes from plastic, has been suggested to have obesogenic activity. Tributyltin, now, probably haven't heard about tributyltin, but it was actually a major component of anti-fouling paints. So near harbors and stuff all around the world, there's lots of tributyltin. And so therefore the marine environment, that is the stuff that we collect from that marine environment, also have elevated tributyltin in there, okay? That's also an obesogen. So there's a number of things like that. Those are the kind of two approaches. So we said, okay, we can't answer everything. Right. So what we're going to do is we're going to propose to basically the Gulf oil spill group, right, the trust fund, that we're not going to try and answer everything, but we're going to be very selective, okay? We know that obesogens are a critical component. We know that things like estrogens and androgens, et cetera, are absolutely critical for short-term and long-term fertility. We know that glucocorticoids, stress hormones, okay, are associated with, for example, inflammation and immune function, et cetera. So we could actually take human glucocorticoid receptor, alligator glucocorticoid receptor, whale glucocorticoid receptor, fish glucocorticoid receptor, and we can line them up in different cells, and we can test these chemicals all at the same time. So not only can we actually say, is this chemical potentially interacting with the receptor, okay, that's associated with stress and in some cases immune function, okay? But we can say, does that cross species? Is it only something we worry about in fish, or does this go from fish to humans? Second part is, because we actually have iPS cells from multiple species, we can do the exact same thing. So we can take the same compounds, put them on these pluripotent stem cells, and say, does it only cause the whales to make more fat cells, or is it actually working in multiple species? And that's something we have to be worried about. The neat part about this is that we can do that in a laboratory, but because other people are being funded, and because we actually also work in one of the major labs in the country for analytical chemistry, that is the Hollings Marine Lab, and the NIST group here, once we identify some of these chemicals, we can now either go back to archive samples, or we can actually collect additional samples and actually say, how frequent are these contaminants found in, let's say, dietary items, or in people, or in dolphins, or whatever that are living in the Gulf. The second part is not only how frequent, but at what concentrations. So what percentage of the population is being exposed, and how much do they have in them? And is that something that we're concerned about? So we can start to translate these findings into, I hate to use risk, because risk is a very specific term in medicine and in toxicology, but you can start to get some idea of whether you should be concerned and whether we need to do more work. So that's the element of the if you will, the BPA Gulf Trust grant. Thank you. And she was right. It says Rachel Carson was right. And I made sure that that quote is in this paper. She took a lot of grief, but she was right.